Hi everyone, Hassam talking from the Sunshine State. I hope you can hear me and see the slides clearly. So today I will share with you some preliminary data we obtained regarding the role of phospholamine. Just as a reminder, we started with this question in mind: phospholemon-mediated regulation of sodium pump. Is it isoform specific? Uh, so in my last presentation, I briefly reviewed what are known about phospholemon regulation of pump, or what are consistent findings and some inconsistent and contradictory findings as well. I briefly summarized the finding of our paper on phospholamine and tried to show that we did not address the question I'm asking now. So this new project would be an addition to the previous work. I also listed what type of stimulation was used in phospholamine studies I found in muscle literature, and also what was measured, trying to show the gap in the literature. Uh, These are the list of questions we might be able to answer once we know the isoform specificity of phospholamine regulation. I briefly reviewed our muscle contraction data regarding adrenergic effect, which is I will explain would be relevant to the current project as well, uh, and others finding. And I suggested a set of experiments designed to address the phospholamine role in alpha 1 and alpha 2 isoform regulations. Okay, and and I also suggested creation of a new mouse model by crossing swap and phospholamine knockout, which turned out it may take much longer time than I would thought, but that's fine. We can do without it, hopefully. Okay, let's move on. And finally, the significance of the current suggested studies. Now let's move on to the real thing, which is preliminary data. So what is shown in these graphs is the force generated by muscle over time. The y-axis is force and x-axis is time in seconds. These are dissected ideal muscle treated with different conditions and then stimulated at 60 Hertz over 1 minute. The colored lines represent the average force developed by different groups of muscle. So overall, the upper the line is the performance of that sample group is stronger. And before explaining the graphs, I want to say that these graphs are combination of data we measured for salbutamol project previously, used here as a control or baseline, plus new experiment with phospholamine knockout muscle. So these are combination of both. On these panel here at the left hand side, these blue lines represent muscle performance after high potassium stress, these lines. Red is the red line is muscle contraction after treatment with salbutamol. And these are, as I mentioned before, these are wild type muscle. So clearly, when we have both alpha 1 and alpha 2 plus phospholamine in wild type, the recovery effect of salbutamol is considerable. The difference between blue and the red line. In this right figure, we have same protocol applied on Poshpholemman knockout mouse. Again, blue is stressed muscle and red is recovery due to the salbutamol. And as you can see, the recovery effect of salbutamol is reduced in Poshpholemman knockout mouse. Again, the difference between red and line, red and blue. If you look at these arrows that highlight the effect size of salbutamol, it suggests that phospholamine is important in transition of salbutamol effect to the pump, because here we have phospholamine, here we don't have phospholamine, and the effect is reduced. Now we have some data that might answer the questions: does phospholamine plays any role in total pump regulation? And the answer is yes. Now here things gets much more interesting. So in the left panel we have wild type muscle treated with wobin, which gives us alpha 1 only activity. Blue again is stressed muscle, and red is recovery by adrenergic stimulation. And as we discussed before, the effect of salbutamol on alpha 1 is very significant. But when I did the same protocol on Poshpholemman knockout mouse, as you see in the right figure here, the effect of salbutamol completely disappeared. There is almost no difference between red and blue line. In other words, when we have alpha 1 plus phospholamine, which is the case in wild type, the recovery effect of salbutamol is maximum. But when we have only alpha 1 without phospholamine, there is no adrenergic recovery effect. So based on these preliminary observations, it's going to be reasonable to hypothesize that phospholamine is required for the adrenergic stimulation of alpha 1. And probably the phospholamine adrenergic effect on alpha 2 is less strong, but some further experiments are needed to suggest so. I also did some rubidium uptake experiment on the same muscle. We did contraction on it, but unfortunately the results had a lot of outlier, which is due to contamination. So it was a little bit confusing, and I decided to wait until we receive results of my more recent experiment and combine all of the data to see what's going on here. And I expect to get the final part of the results from Julio's lab by today. If everything goes well, hopefully, I will present that part of the data next week. So that's all I had for today. Thank you. Bye.